This conference will now be recorded.
Welcome everybody to the webinar on high tunnel tomatoes. Today we have Becky Maden with the University of Vermont Extension, Nutrient Management Specialist, and Cheryl Sullivan from the UVM Entomology Research Lab. They're going to show you quite a number of slides and go rather fast. All of these slides and the recorded video will be posted on the Vermont Vegetable and Berry website, so you can review them later if you missed something. And we'll ask that you put your questions into chat while they're presenting, and then we'll review them at the end and also have time for verbal Q&A if you'd like to ask questions or offer up some experience that the group might like to hear. With that, take it away, Becky.
Thanks, Fern. So yeah, I do have a lot of slides, but just so many fun things to share and show about high tunnel tomatoes. And I'm super excited to have this group of you on this today, and I hope we leave a lot of time for questions. Feel free to type them in the chat as we're going, but as Fern said, we'll take them towards the end so we can cover everything. So Cheryl and I are just covering two of the many elements that make high tunnel growing successful. There's a whole host of other folks in extension and in the ag community working on other aspects like ventilation and humidity. So this is a narrow window, but hopefully a good start for high tunnel growing. You all know why to grow in a high tunnel, but just to emphasize that it's a pretty awesome addition to our agricultural landscape in terms of versatility and protection from the elements. And when I started farming not terribly long ago, this is what high tunnels looked like. Pretty small structures and pretty simple, pretty low investment. And we've upgraded, right? They're getting taller, they're getting bigger. We've got some NRCS funding, which has helped us build more. And now we've got growers growing in ground in tunnels like this, which are significantly expensive. They're quite beautiful to work in. They're high, they're well ventilated. They've got a lot of automation and they're expensive. So what I'm really trying to get at with my work is how to address the needs of growers who are in this wide range of tunnels, and also optimizing the yield coming out of them. Because when you're investing any amount of money and labor into these structures, you have to make sure that you are recuperating the costs. So this is kind of the angle we've been taking, is like how to address this range of tunnel structures and also crops. So we're doing a lot of winter greens, cucumbers, ginger, but ultimately it comes down to tomatoes, right? They are kind of the tried and true tunnel crop. They are generally pretty profitable. You can put a high density in the tunnel for a long period of time. We've got a pretty good understanding of how to grow them, and Vermonters love their tomatoes, right? So most of the work we've been doing has focused on tomatoes, and that's kind of our starting point for honing in on soil fertility in high tunnels. Now, we're also talking specifically about growing in the ground in high tunnels. We're not talking about hydroponic growing. A lot of growers are either certified organic or using organic practices in tunnels. So that means a pretty dynamic system, and it's really tricky to address. There's so many variables, so many different native soil types we're working with. And we also have different crop yields that we're talking about here. So this is my colleague Dana Rupert taking a soil sample in a tunnel this spring. And we've got these like massive plants coming out of these tunnels. So how do we balance these two things? You've got your soil test, but then you've got your expectations of what's going to come out of your tunnel. And we've also been doing more and more with kind of manipulation of the soils. On my farm, we have really heavy clay, so we've been playing around with raised beds, trying to reduce the compaction in our tunnels. I also like, there's a lot of other things we liked about the raised beds. So, and again, we've got this like serious heavy nutrient demand in a lot of these tunnels. And if you just think intuitively about the biomass coming out of here, this is one farm I worked with. They were measuring every week how much the plants were growing. So that's the first, April, what is that? I can't read April 20th to 27th. and then April 27th to May 4th, and then May 4th to May 11th. So much growth each week, right? That's an incredible amount of nutrients that the plants are demanding. And another visual of that is these monthly crop images, how much biomass these plants are really accumulating every month right through November. The incredible nutrient demand. So to jump right to it, because we don't have a lot of time today, therefore, we need to tailor our nutrient recommendations to these crop yield potentials. And this chart is part of a larger fact sheet that Vern and many others in New England put together as the result of a study we did in 2018. But you can see that there's not one blanket recommendation for tomatoes in a high tunnel anymore. We are trying to really refine it, and we're continually doing this based on what the yield expectation is of the crop in the tunnel. So a lot of farmers have said to me, well, nobody wants a low yield, but when we're describing a low yield, we're actually talking about maybe a short season determinant. You might plant a late determinant tomato and just want to get one quick harvest and be done and be out of there. So you're not putting on a lot of biomass, you have a lower yield. High yield might be a grafted hybrid that you're planting in February or March, and you are hoping to harvest right through October or November. You probably have supplemental heat in that scenario. Most of the growers we work with are in that good yield range. So looking at like 120,000 pounds of fruit per acre, which is awesome. That's so much fruit. And one thing I really want to emphasize, we're talking specifically with most growers, honing in on nitrogen and potassium. And potassium is really, really the nutrient that seems to be the most elusive in high tunnel growing. It seems like we can't quite get enough to the tomatoes no matter how hard we try. And you can see these numbers are huge. Compared to a field, the potassium tomatoes demand is really intensive. So when we're talking about soil nutrients, we're not talking about a discrete fraction that's available in the soil. We're talking about what would be available to a plant over the course of its growth. So a field soil test, modified Morgan's, as you can see on this chart, is what we use in New England. And that is a soil test that tries to simulate the conditions, the field conditions in New England on a typical growing year. So it's a weak extract, it pulls out nutrients, and it's a predictive test of how many nutrients or how the amount of nutrients a plant could access over the course of that season. That's kind of the best thing we have for field soil tests. That's not what we're seeing in a greenhouse in terms of growing conditions. So we've coupled that test with a saturated media test, which is primarily used in like greenhouse media, potting soil media, and it shows how many nutrients are available immediately under, like, it's a water soluble solution that they use, a water extract, and it shows how many nutrients are available right off the bat. So using these two tests together helps paint a more complete picture of what plants can access in a high tunnel situation. The saturated media test also gives us important information about salts in a high tunnel because we're not getting the rain to flush the salts away. We're also working a little more with leaf analysis, which I think is becoming a more valuable tool, and that can tell you what the plant is actually taking up. So you have your soil test, but you don't always know what the plant is actually doing with those nutrients in the soil. We really like working with UMaine. I would highly recommend using their long-term tunnel test. They've been a little slow this year with COVID because other labs were shut down, so it's been about 2 weeks. Tissue testing, therefore, is not necessarily super useful for turning the ship around during this season, but I think it's a very useful tool for at least knowing what's happening and kind of modifying next year. It's become a great research tool for us to understand how the plants are behaving in conjunction with the soil tests. Dairy One, I think, has a quicker turnaround. If you have like an emergency tissue situation, you might use them or call them and see how quick it is. This is what the soil tests look like from the University of Maine. This is the modified Morgan field soil test. What I want to emphasize quickly here is that Bruce and the lab there have done a great job with the recommendations. So if you have no time to talk to Vern or myself, what I've circled here in purple is he's using the saturated mead test to provide these recommendations. So I would use, these are great. They're right on, spot on. The other thing I wanted to emphasize is I've circled, this is one of my old tunnel tests. The potassium here you can see is above optimum. The reason why we want this saturated media test is because you can see the available potassium is actually quite low. So that's giving me the information that, yes, I have plenty of potassium for my crop for the season in that tunnel, but I don't have enough maybe to get that plant started, that jump start right away. I'm putting in this huge, beautiful 4-inch pot tomato plant. It's ready to go. And if I don't have the nutrients for it right off the bat, it might struggle a little bit. So this is why we use these two tests. I also want to emphasize that this is my job and Vern's job is to be able to look at these tests and help you, so you don't have to struggle with figuring out what the green and pink numbers, the alarming high numbers, what that really means. But they're useful tools. Just quickly, some of the soil amendments we like. I really like the seed meals for nitrogen. I'm going to blow through some other pictures of those. We're working more and more with fertigation. So if you have the capacity, I think, or want to invest a little bit of money, this is a great, you can either plan on it or just use it as a backup if you're seeing problems. The New York folks working on high tunnels recommend putting 1/2 to 2/3 on of the nitrogen and potassium right up front, and then doing these weekly doses. So, you know, sometimes when I've had problems and didn't have an injector, you can just kind of dissolve the nutrients in a bucket and water them on. But the Mazzei Injector is like $100, and it's pretty straightforward. You dissolve, we just dissolve sodium nitrate and potassium sulfate fines in a bucket, and you can just put it on. It's not super scientific. A dosatron is much more precise. Soy meal, I love it. It's slow release. It does a great job in the tunnel. My only warning on that is the rats really love it. And if you don't give it the true window of like 2 weeks before you plant, I have seen it kind of heat up and cook the stems of plants because you're putting just so much in that tunnel. So ProGrow, tried and true, people love that. This is the sodium nitrate that dissolves well in a bucket. You need warm water and agitate it a tiny bit. Bone char is the most available phosphorus source if you need that. These potassium sulfate fines on the left-hand picture are a game changer, I think. They dissolve really easily. I know one farmer who just puts them in a bucket the night before she's going to fertigate, and they dissolve overnight. They're very readily available to the plant. So for that potassium issue, those are great. And like Laws Ag and a couple other suppliers in our region carry those. Epsom salts for magnesium, gypsum if you need calcium and it doesn't change the pH of the soil. Peat moss has also been a bit of a game changer. Heavy compost additions over time have led to an increase in pH in many tunnels. Peat moss is a good alternative because it mildly acidifies the soil, but it also adds organic matter. So this is a farm that buys it in bulk. They're awesome, huge marshmallows that are incredibly heavy. And this is the rate that typically we recommend. Sulfur, we're seeing more and more tunnels that have been in production for a long time with the pH going really high. One of my tunnels, this is super embarrassing because I'm supposed to be good at this stuff, but one of my tunnels was 8.5 pH this year. Can you believe that? So the sulfur takes a long time to activate. So keeping an eye on that pH rather than letting it get above 7.5 or 7.3 is really important. I've also been seeing, you may have seen this on your tests, the sulfur levels getting really high at the same time that you're trying to acidify. And I think that's a microbial activity thing. So really keeping the microbes healthy, keeping the soils moist to get that sulfur activating. So yeah, another place to wear your mask is in the tunnel when you're tilling. Salts, not so much an issue with tunnel tomatoes in terms of growth factors, but when you finish a round of tomatoes and you go to get your greens in, you might see this. That is salts accumulating. They tend to build up on that top 2 inches of soil. So finding some way to incorporate or flush the salts is pretty critical if you're following your tomatoes with greens. And just quickly, I just want to acknowledge that this study we did in 2018 has been the foundation for the work we're doing now, and that was a collaboration with all these other folks in New Hampshire and Massachusetts, Maine, Rhode Island. The study had 20 farms, and I just wanted to show this to show the vast array of yields people are getting, and it comes back to that, like, got people growing in a million different ways and lots of different size tunnels, different scales, but we're still kind of above that field range. And just quickly, there's a lot of other things going on, and this is why we have Cheryl and Anne and Chris and Andy and Vern, everybody working on this. But hornworms, these are the other problems from that study. Powdery mildew, Anne talked about some of these things last week, but that's hasn't shown up this year. Botrytis has been a big thing this year. I've been seeing tons of it, especially blossoms, leaf mold. And this was just a picture from last week, tons of heat-related issues. Irrigation's critical. This farm has 4 lines. They turn on 2 early on in the season, and then they do 4 when the plants get bigger. It's a great system. Leaf removal, humidity, and all sorts of tricks and tools you can geek out with. Super fun things. I was hoping to get a picture of those stilts in action, but I didn't. And this slide, we're not going to read through right now, but I want to leave this up on the website for folks to check out. So, and we are doing this project this year. We would still welcome growers to join. Sorry, I didn't know there was all that, but we've almost completed 1 year of it, but I would welcome any more growers. You know, most of you guys on this call probably aren't actually growing tomatoes right now, but that's it. And I'm going to switch over to Cheryl.
Hi, I had just sent the request to share the screen.
I'm on it.
Oh.
Okay, you should be good to go, Cheryl.
All right. Hi, everybody. Thank you for inviting me to talk today. Can we see it? Oh, there it is. It's coming up. Can people hear me?
Yes, sounds good.
Okay, good. All right, fabulous. Awesome. All right, so I'm going to talk about tomato IPM from the bug perspective. Why is this not working? Oh, there we go. Okay, so just a quick recap in case you missed Margaret's discussion last week about cornerstones of IPM. It's a decision-making process with a foundation based on prevention of pest problems, where you're going to use chemical pesticides as a last resort. So there's a strong emphasis on scouting to identify pests, to determine where they are, what magnitude on the crop. You should develop thresholds for the given pest to determine how much damage is tolerable before they affect the current and future value of your crop. So with this info, you'll be able to make your management decisions based on your own personal cost-benefit analysis. And I'm often asked what strategies are best, but there really isn't a cookbook approach to IPM. And the programs that you develop, they're uniquely tailored operation to operation. So really, today I'm going to focus on pest prevention strategies and the use of the cultural strategies and biocontrol agents. Throughout this talk, there's also many links scattered about for more information on certain topics. So within your IPM program, scouting for pests is critical. It's one of the most important things you can do to prevent the outbreaks. It's essential to determine what the pest is, how many plants are infested, the magnitude, information such as number or percentage of infested plants per bed or tunnel, or the use of a rating system of number of insects per plant or leaf is really critical information that's needed if you plan to use biocontrol agents. Usually, if you order biocontrols from a supplier, they need to know that information as well as tunnel size, and those magnitudes of pest pressure. So there's often times where if there's too many plants infested with such high numbers of pests, the use of biocontrol is not going to be a cost-effective choice for you. Biocontrol works the best as a preventative and it's not curative. So if the level is too high, you might want to consider some pesticides instead. So your extension agents or a biocontrol supplier can help you determine whether or not biocontrol will be the right option at that point in time. So you also need to identify what stages of pest you have. Some natural enemies only attack immatures, while others may attack both the adults and immatures, and even the eggs. So I can't emphasize more the importance of keeping records also. So you want to write things down in a notebook to refer back to what worked, what didn't, why might not it have worked, were there too many pests, was it the time of year? Oftentimes it takes a lot of trial and error to develop an effective biocontrol plan that truly is unique for your own operation. Okay, so with all that said, let's start to discuss some common pest problems you might encounter on tomatoes, starting with mites. These are namely two-spotted spider mite, broad mite, and the tomato russet mite once in a while. So let's start with the ones that you can actually see, which is two-spotted spider mites. They tend to have the two dark spots, and you find them on the leaf undersides. It's important to remove your weeds in tunnels if you plan to leave it fallow over the winter. These mites like to overwinter in debris along the tunnel edges and like the piping and structures or wood along the edges, foam, anything. They can turn red during the short days of winter. Often infestations usually start along tunnel edges or near places where they've overwintered. Broad mites are something that have been showing up more and more the past several years, and I've been seeing them pop up quite a bit in tunnels, so I want to spend some time discussing them. I tend to see these a lot in locations where transplants were obtained from places that were also producing greenhouse ornamentals. These mites can really dramatically affect peppers, eggplants, and tomatoes as well. Unfortunately, once you see the damage, it's often too late because they're microscopic, so it often goes unnoticed. So if you see the damage, it's best to pull out any infested plants, especially when things start fruiting, because yields can go down quite quickly. These mites can move throughout the tunnel on other flying pests like whiteflies or even aphids. As these mites feed, they release a toxin that actually has a growth regulator effect. So at really low densities, this pest can cause significant damage. So if you use things like pesticides or something and you spray for them, if the mite's been controlled, you can actually keep seeing damage appear as a result of that toxin. So these are tiny. They can only be identified with a microscope. They actually have some really cool looking eggs with that mottled appearance. Really though, I see them most on peppers and eggplants, but I've seen them jump ship to tomatoes if they go unnoticed. Also very microscopic is a tomato russet mite. Occasional problem, another reason to remove debris. These can also persist on other solanaceous crops because they can use them as alternative hosts. So not all mice are created equal. If you decide to use the pesticide route, if you notice these mites all belong to different families. And the reason why miticides aren't universally effective is because of their life cycles and behaviors. So miticides that control spider mites may not be effective on the others and vice versa. The good news is with the use of natural enemies, there are many generalists that have better efficacy across the species. So what does some of the damage look like from these mites? A lot of it's similar, a lot of it's different. Broad mite, you tend to see severe foliar distortion and curling at the tips. That is usually a dead giveaway that it's the broad mite. And the russeting on the fruit, you can also get splitting. And it's really the leaf curling and distortion that is a dead giveaway. You just kind of come in, you're like, oh, what happened to everything overnight? Tomato russet mite, this is something, the damage usually shows up from the bottom and goes up. So usually it's russeting on the stem, and in severe cases it gets to the fruit. And oftentimes it gets this powdery appearance, and That's actually colonies of mites. There are tons and tons of mites that just kind of stick together and look like a powder. And then you get the foliar distortion and the curling of leaves. And spider mites, which I know we're all familiar with, they tend to have that mottling appearance, the stippling on the leaf surfaces, and they get the webbing. When things get severe, they get the yellow flecking on their fruits. And I hope nobody ever sees a spider mite infestation like on the plant in the right-hand corner. That's actually an eggplant, but I saw that at one site visit and was just really blown away by the fact that there were actually spider mites dripping off the plant. I hope I never see that again. So you want to avoid that. So how can we avoid that? Fortunately, all these mites can be very successfully managed with biocontrols before they reach the spider mite dripping level. A lot of the mites in the Neostilus or Amblicius genre are very effective mite predators. These are all very tan in color. I can't tell them apart, even under a microscope, so I wouldn't expect anyone to be able to in the field. The good thing about the mites in this genus is they also eat pollen in the absence of prey, and a lot of them are available in hanging slow release sachets, where you hang them on the plants, which allows them to persist for a longer period of time in the crop. So for spider mites, there's actually a predatory midge. It's a fly, it's larva, it's a maggot, it eats the mites. Adults feed on nectars and pollens. There's also Swirskyii and Andersonii. And then there's a specialist mite, Persimmilus. Persimmilus only eats spider mites, and it actually does a really good job at clearing spider mites out when populations are below damaging levels. These, however, won't persist without any prey, unlike the other mites. So in spring, I've seen growers have really good success using the soil predator Stratiolaelaps scimatus. I can't believe I just said that right. Formerly known as Hypoaspis. You can purchase those and release those along the edges of the tunnels in the spring, and they tend to eat a lot of the overwintering mites. This is actually a generalist mite. It actually works well on thrips as well in the soil. So for tomato russet mite, there's Phalaensis and the Andersonii mites, and for broad mite, you typically use Cucumis and Californicus. So all these mites have different ranges of like humidities and temperatures that they do best. So it's really important to use them at the time of year they're going to be the most effective. That's why they give you a couple different choices, because one might do better in early summer as opposed to late summer, just based off their heat and humidity tolerances. So what else can you do? Well, from a cultural perspective, other than removing debris and everything, you can use bush beans as a trap plant. These work really well when they're young, planted along the edges of the tunnel or scattered throughout in the understory. They're a great scouting tool because the damage shows up easily on the smooth leaves. And they also grow really fast, and they can be replaced readily throughout the season. So they're great for a trap plant and for indicator plant to actually indicate that you have a problem that might get out of hand. So let's talk about thrips. Thrips are obviously concerning due to their ability to vector viruses that can decrease yields if not managed. Viruses can be transmitted by numerous species, but up here we're mostly concerned about western flower thrips and onion thrips. We see those the most, especially the latter. If you grow onions, they can get pretty nasty. I usually see thrips infesting tomatoes early in the season when herbs or eggplants and cucumbers are produced along with tomatoes in tunnels. So if you have a mixture of crops, early it's largely due to the plants being produced in areas where there's other infested plants, like transplants from other folks who also produce ornamentals if you buy ornamentals in, kind of like the broad mite. The broad mite likes to hitchhike in on the greenhouse ornamentals. So both these species lay their eggs in plant tissues, and they pupate in the ground. So the ground portion of their life cycle is often overlooked for management. So you need to account for the thrips that may be emerging from the soil, even if you don't see them on the foliage. There's many different stages of the thrips larva that vary in shades of yellow, and you can find these on the undersides of the leaves. So onion thrips tend to be found mostly along main leaf veins, where most damage is evident, where these veins kind of come together, where western flower thrips tend to be more scattered, again, on the leaf undersides. So some of the damage, they damage plants by piercing the cells and sucking out the contents, and it causes the tissues to die. That's where you get the silver gray patches on the leaves and the characteristic black dots of their excreta, and that usually indicates their presence. These reduce the plant vigor because you lose the chlorophyll, and of course viruses might be transmitted, which would affect the quality and yield. So as far as natural enemies, there's lots of options for thrips, ranging from predatory beetles and bugs to mites. It's best to combine strategies that get both the foliar and the soil level. For example, the roe beetle and the Stratolelaps mite, they're soil predators, and they need to be combined with the foliar-dwelling mites like cucumerus or the predatory bug aureus. Marigolds can act as a fabulous scouting tool to help detect thrips early in the season. Thrips are attracted there, and you can tap the plants over a white sheet of paper to monitor their presence. Or you can hang sachets of cucumerus onto them to provide added suppression. Sometimes you can put a sticky card on them for additional scouting power. So if infestations get too high, you can just bag them up and throw them away. The key is to have them in flower early in the season, really when tomatoes and your other crops are first planted or started. So you need to plan ahead to use marigolds often before you even think about putting stuff in the tunnels. All right, let's talk a little bit about whiteflies. Two species in tomato. The biggest threat is the greenhouse whitefly. Both can vector viruses. However, silverleaf is more associated with ornamentals. So if you find a silverleaf, it's because it came in somewhere from infested stock. The difference between the two adults is really subtle. The silverleaf have tent-like wings that, where the greenhouse whitefly holds them more flat. The silverleaf whitefly nymphs are pancake-shaped, or like scale, I guess, scale-shaped, whereas the greenhouse whitefly is more cake-like and tends to be really hairy. And both of these can be effectively suppressed using parasitic wasps. So Eretmosceras is more specific to silverleaf, whereas Encarsia is for greenhouse. They will attack the other species and vice versa, but each one is more effective on their preferred whitefly host. So they're often sold as mixes, but straight Encarsia would be best for tomatoes. They come in a hang tag form. And usually you can tell these are working because the wasp parasitizes the immature whitefly nymph. And with Encarsia, the nymph turns black, like a black color. It means it's been parasitized. Whereas the Eretmosceras, when it attacks the silverleaf, it turns a more yellow gold color. So it's important to know these wasps only target immature stages, not the adults. And here's some signs of whitefly damage. And usually with the whiteflies, you tend to see leaf yellowing and chlorosis from their feeding, as well as you find a lot of sooty mold from their excreta when infestations get really high. Okay, the Aphid Apocalypse. Aphids definitely are the #1 pest in high tunnel crops in the Northeast. So our surveys have said. Some may argue that. Aphids are one of those things that just don't go away. Time and time again, I see growers rotate crops in and not manage existing aphid issues just to have an explosion occur later on down the road. Again, if you're fallowing tunnels over the winter, remove all the debris. I've seen one tiny weed be covered with aphids over the winter, just waiting for new stock to be planted or seeded within. It's kind of crazy, just the way that they can survive in tunnels. So on tomatoes, the aphid of concern is a potato aphid. with foxglove a close second. For growers growing winter greens, you're going to likely have green peach aphid on those greens. And often when transitioning over, these will jump from the greens to the tomatoes. And infestations can look really scary, but after pruning and use of biocontrols, they seem to fizzle out over time. That's not the case with potato aphid. They'll persist and absolutely devastate a tomato crop if left unmanaged. So be sure to check your plants if you buy them in before planting in the tunnel. I tend to see foxglove aphids hitching rides when plants are passed along to new locations. They tend to be a cooler season aphid and can overwinter more effectively than the others. So they've been doing really well here over the past decade. So it's good to know what species you have, because the biocontrol agents that you're going to use are often specialists and they attack only certain species. It's challenging to tell the aphids apart. You really can't base it off color, and really the head shapes are subjective. But really though, if you see large green or red aphids in the middle summer on tomato, it's likely potato aphid and you should seek help for that. Foxglove aphids have 2 dark spots at the base of the cornicles, and that's really easy to see with a hand lens. Both the foxglove and the potato aphid tend to be a larger aphid in general. Green peach, that's present if you have things like eggplants, greens, or peppers. Oh, they love peppers and greens. Well, they like a lot of things. But really, melon aphids often show up a lot on cucumbers, and melon aphids have really distinct, dark, blackish color cornicles. I hope no one ever sees the potato aphid infestation on their tomatoes like that picture on the right. That was a very scary infestation that resulted in very high loss of crops. So their damage, cast skins, that's a telltale sign. They're white and they blow around. Often they can be mistaken for whiteflies. Again, they're on the undersides of the leaves, but they can show up on the tops of leaves because they fall down and just kind of rest on the surface. Distortion's quite common, and honeydew with the sooty mold as well. Same thing in the picture on the right. There's a lot of moldy tomatoes as a result of a really high potato aphid infestation. So some of the natural enemies. Again, know your aphids if you use the wasps, because these wasps tend to be specialists. You have Aphidius colemani for green peach and melon aphid. Those can be reared using the aphid banker plant system. I'm not really going to talk much about the aphid banker plant system just because it has a lot of potential for work in high tunnels, but it takes a lot of time and dedication and playing around with to make it work effectively for you. Aphidius urbi, that's a larger wasp, and that attacks potato and foxglove aphid. There's Aphilinus, that's a small wasp, and it attacks potato and foxglove aphids. There's also a lot of naturally occurring species that come in, and they parasitize the aphids, and they turn into the mummies. So the Aphilinus has black mummies, and the Aphidius has brown mummies, and all other native species have all sorts of colors of mummies. But the preyons are pretty cool. You might have seen those. I see those quite a bit in high tunnels, and they actually have the wasp is actually developing underneath the aphid. You can see that's got a really, it's like an aphid sitting on a pad. They're actually quite cool when you see them. Things like lacewings, those do a really good job. And the predatory fly, Aphidoletes, that's a generalist, and that has really great potential in high tunnels to knock down aphids because it attacks all species. It does well with dirt floors. Again, it's predatory as a maggot, and its adult feeds on pollens and nectars. So you might notice that I didn't have lady beetles on here. Those are better suited for aphids, in my opinion, on winter greens, early season or late season, going into the winter. Usually when you use lady beetles during the summer, they have a tendency to just disappear. But during the winter, if you can put them under row covers or greens, they tend to have a better chance of sticking around to do their job. All right, hornworms. I'm going to talk a little bit about hornworms. It's a quintessential tomato pest. The 2 types of tomato hornworm. with the white V-shaped stripes on its body and a black horn. We see this one a lot later in the summer, but really it's a tobacco hornworm is the one that we see the most of. It sports the diagonal stripes and the red horn. So they can be seen earlier on in the seasons if they actually overwinter in high tunnels, because they do. They pupate in the soil under the debris. So they can get really large, obviously. They do blend in with the foliage, so it's kind of hard to find them at times. So for scouting purposes, I just don't really look for caterpillars. I just look for the frass on the foliage, as it's easier to see than themselves. They often tend to feed higher on the plant, so I tend to look up. So regardless of which one you might have, they can be easily managed with well-timed applications of BT bacteria, targeting the early stages of the larva. So the BT occurs naturally, and it's a rod-shaped bacterium that produces spores and crystalline proteins. So the worms actually need to eat the proteins in order to be toxic. So if you're using BT, you want to read the label to make sure that if it tells you to do repeat applications, that you follow that advice, so you can actually get the feeding stage. There's also a lot of commercially available and naturally occurring parasitic wasps that attack caterpillars in general, and the BT is pretty in general as well for a lot of other caterpillar issues. So there's trichogramma wasp egg parasitoids, and there's actually the naturally occurring Catesia wasps that lay their eggs in the worms, and then they come out and pupate above it. I'm sure you've seen that before. It's pretty cool. All right, I threw this slide in here. I'm not going to play it because the videos can be a little graphic. I don't want to have a huge freak out by watching it, but these are really awesome YouTube videos from National Geographic, and one of them is parasitic wasps versus aphids, and the other one is versus a caterpillar. They're really awesome, and they're really worth checking out if you're interested to see how parasitoids actually affect their host from the inside out. So what else can you do to prevent pests? As a part of an IPM program, you want to think of providing suitable habitats for your natural enemies that you purchase, or even natural ones that you want to attract, the freebies. Often a lot of the adults of many of the parasitic wasps and flies, they need the pollens and nectar resources in order to lay eggs and effectively find their hosts. If you get things like high tunnel crops, there might not be a lot of pollens and nectars available in things like tomatoes and especially cucumbers. So providing habitat plantings can be a huge help. So the habitat plantings, they provide the food and resources as well as attractive pests to beneficial insects. So like habitat plantings of alyssum, marigold, bush bean, and high tunnel tomatoes. That we've done some research with that, and we've had really good results showing a lot of different natural enemies. They tend to attract a lot of syrphid flies and other aphid parasites. So the flies actually look like bees, and they're actually really awesome pollinators as adults, but their larvae are the ones that consume the aphids, and they're free, which is really awesome. But they also attract other things like lady beetles and a lot of other things. So it's good to plant habitat plantings if you can. So here's a bit of a breakdown of what we actually saw on our habitat plantings. We saw a lot of parasitic wasps. There are mummies too, because sometimes we use a few other different plant species like borage. We don't recommend the use of borage because borage attracted everything under the sun, including aphids. So there was a lot of parasitic wasps and mummies that came to the habitat plantings just because the pest was there. And then you have something like Aureus. Aureus who you can actually purchase, but they actually come in from the outside later in the summer. They really like yellow. They really like marigolds. So if there's any thrips around, they'll feed on pollens and nectars, and also any thrips that might be attracted. And of course, you have the syrphid flies. These are the primary things that we saw. But there's also things like lacewings and assassin bugs and all sorts of other cool things show up on these. So I didn't really talk about pesticides just for the sake of time, but they're an important part of your program when your other strategies might have failed or things got overlooked. We just didn't have the time to scout and find your pest problems. That's why it's good to designate somebody to scout to find these problems, to get at them before they reach a level that you might need pesticides. But in case you're not aware, for anybody that's new, I really like table 21 of the vegetable management guide. It's got a great listing of products broken down by pest. Also that link for insect control. It just gives all sorts of listing of what you can use. I always feel the need to say that just because something is branded as least toxic or biorational doesn't necessarily mean that it's soft in the non-target effect department, because a lot of products can still be broad spectrum and affect natural enemies. So on that note, there's a lot of locations where you can get info on protecting beneficials from pesticide applications. There's a really cool article by Cornell that came out earlier that has a good listing of compatibilities or links to places where you can find compatibilities. Also, your biocontrol supplier really should be your main person to go to ask these compatibility questions to. So if there's anyone that needs certification or use of pesticides, you can talk to Anne at the Pesticide Safety Education Program that they run here on campus. So here's some more additional resources about managing pests in high tunnels. We do have a high tunnel pest management webpage. It's in, what is that? Yellow, orange. Up top, or third one down, that has a bunch of links to a lot of the research you did on habitat plants and other resources to this area. So these are fun to check out. So that's all I have. I just want to mention that there might be a high tunnel conference, the third one, in December. It'll be virtual. Details will be coming out on that probably shortly, I would assume, but that's something coming down the line. So thank you very much, and I see you I have 10 minutes for questions. Is that right?
Yes, let's open it up to both Becky.
Hello?
We're still here. One question I have, Cheryl, is there a list of biocontrol suppliers somewhere that are serving the New England area?
On our website, we have a link to a lot of different biocontrol suppliers. There's so many now. But even with the ones that you choose, it's good to feel that you can build a relationship with them and be able to talk with them. Because there's a lot of places you can buy stuff online, or you just order it over the internet. But really, I like to find those places where you can have that one-on-one consultation, because they're ultimately going to set you up with programs, because not all suppliers are created equally, and some will help you more than others, and some even have reps that can come out to your farm to help you with those plans. But I'll link, before I send that, I'll link to that.
Great. And I know some of the suppliers are Canadian. Is there any recent problem with getting biologicals, beneficials across the border? Or
I've heard things have been running smooth. I think a lot has to do with production and having things be produced on time. Because it's a struggle. They're live things and they're prone to population crashes. So sometimes you order something and it might not be available because something went wrong, or who knows, or holdups at the border. But things seem to be running smoothly, unless anyone wants to say differently.
Any questions from the group? Well, I have another one that's at the interface of both your presentations. I mean, it's been kind of common knowledge that excess nitrogen promotes aphid feeding and aphid problems, but I don't know that I've ever seen any research that actually substantiates that. Is that something that you think is true?
Well, there has been research done on that, but things change. 10 years, 20 years. Those are things that should probably be revisited in different areas or in high tunnels, at least in our area. So, but yeah, they say that is what happens.
I mean, I'd say to that, Vern, too, just anecdotally, like without any research, it seems to me that with tomatoes anyway, they really like the new young growth on them, like the young plants. And like Cheryl was saying, I think with the, if it's not the potato aphid, they kind of dissipate once the plants grow a little more rugged. So I don't know if it's the high nitrogen per se or just the fresh tissue that they like. And I don't know, that's just anecdotal.
There still is aphid management in high tunnels. It's a struggle. It's a challenge. And there's a lot that we don't know, and there's a lot that should be researched, and there's a lot of opportunity there, in my opinion. So.
How about the break between tomatoes going into winter greens? Do you have some sense of if that's important and how long it should be that the house is totally devoid of plant matter for aphids before you plant your winter greens?
That's $1 million question, and I don't really know if there is the best time or the time length to let things go fallow. I know a lot of it has to do with what was in there before. Were there aphids in there on the crop then, what the crop was? I know I talked to some growers that don't even want to do winter greens because of, the aphid problems. And I think it has more to do with being conscious of what pests are in there when you rotate crops. Because if you know that you have a problem, you can spot spray with something, you can nip it right in the bud to be sure that you reduce the chances of it carrying over. So it's just really being diligent of knowing what's in there and being on top of things. So yeah, it's a tough one. Because it's different for every person. Growers might not be able to afford to let things fallow, so it's a challenge, and I think it's something that we need to work on and research a little bit to kind of figure out what sort of turnover strategies might be best in the context of pest management.
Any other questions? Well, I have one for you then, Becky, about this transition. I know that you're working on this, and I believe UMass is also trying to understand if additional fertilization is typically needed after a tomato crop when you go into winter greens, or if there's enough residual nutrition. What's your sense of where that's headed?
Yeah, I mean, I think that's a kind of what I'm seeing is the next step in our research to dial in more accurately. But my sense right now is I think it's useful to take, if people can, take another round of tests following the tomato crop. Generally speaking, I think there is ample nutrients for winter greens. But again, I think a little bit comes back to that biomass and crop density understanding too. I think if you're trying to grow a longer season, like a full kale or full chard, you might need a little additional fertilizer. But if you're just trying to do like a you know, 50 or 60 day arugula crop, maybe not so much. I do know a couple growers who started amending with, you know, kind of the equivalent of 30 pounds of nitrogen per acre for crops of bok choy and I think spinach, and felt like they had much better results. But again, I think that's the next step. On the flip side, I have worked with some growers this year who took their tests and then did a round of greens and then did their tomatoes, and some of them have thought that they were a little low in fertility, even though we tried to adjust for that. So I don't know if it's necessarily the quantity or if it's the timing of release. But there is obviously some crop removal from the greens. I think more nitrogen, I think potassium's not really an issue there.
So this interface of the two crops and these two issues of nutrients and insect pest management is pretty interesting. I was just thinking about tillage. So if your salts have moved up to the surface through evapotranspiration, it would seem like tillage before you planted winter greens to mix those salts in deeper in the profile would make sense. But Cheryl, is that a pro or a con or neutral as far as if you've introduced beneficial insects, will tillage, what kind of effect would tillage have on them?
I'm not sure. I mean, obviously you have stuff in the soil complex, and that's a really good question. And I don't have an answer for that.
I do think the tillage thing is something to kind of emphasize, because I think as more and more growers are looking towards no-till, the high tunnel is a little bit of a low-hanging fruit. It's like, oh, I can start my no-till practices in the tunnel, but the salts thing is a really big deal for greens. And I'd say after 3 or 4 years of amending and growing in a tunnel, people are starting to see these salt issues, and I think it's pretty significant. So I sort of urge people to at least monitor their salts if they're heading in the no-till direction. I don't really know what it does for the beneficials, but yeah.
I mean, it's a tough one without you know, researching it and knowing what we would see. I mean, obviously there'd be a greater complex, but then you have a pest complex. So how do you know if it's going to be in balance, and when's it going to get out of balance, and how do you fix it when it gets out of balance, you know?
Well, that's a great note to conclude our webinar on. Lots of unanswered questions, a lot of research to do in the future, and some areas where it'd be great to have grower observation and grower records to share with the community at a future date. So with that, thanks everyone for attending. This will be posted on the Vegetable and Berry website, and I hope you can join us next week when we'll hear from Chris Callahan, Andy Chamberlain, featuring some case studies of post-harvest facilities that they've been working on. Take care, everybody.
Thank you.
Thank you.
Thanks, Vern. Thanks, everyone.